So all of this stuff that's on the board, you need to know.
What is that first word mean?
What first word?
Under CHO.
Under that little delta,
Yeah.
it means different, and 8 means differentiate. Okay, let's make sure we know all this stuff, right? This means to differentiate. That means carbohydrates. Okay? This means after. That means about. ATC means around the colony.
What is after?
MO means microorganism.
Which one is after again?
Post.
Oh, okay.
What's that? Difference. So this means difference and 8, differentiate. That's what that means. Because it takes a long time for, look how much writing that is. So I use shortcuts. Okay, so today the only thing you're gonna do for me in lab is inoculate this media. It will take longer for me to talk about this stuff than it will for you to actually inoculate. But I'm gonna give you all the things you need to know. If you didn't do so well on the practical, it's because you weren't paying attention. I give all of the answers for the practical in lab. There is a matrix of all the tests and all the results, everything you need to know on Blackboard for you. That was a student-made thing. People loved it so much, I asked if I could have it and I just put it on. When you get to the third one, I've done that one. You'll see the difference. Okay? Because I got the idea from her. Anybody have any questions? All right. So we start with the phenol red broth test, or it's also known as the carbohydrate fermentation test. So this is the way the broth looks. The broth, when you're going to test it, is going to look red or orange. That's because it has a pH indicator in it called phenol red. The other part of the setup, because they're seen over there, number 7, setup, is this little tiny tube inside of it that's inverted. That's called a Durham tube, and its function is to capture metabolic gas. Okay? So this is the way it starts out with. When you inoculate it, it always starts out neutral, and then the organisms will do something with the substrate, and they'll either turn it to acid, they'll make acid and gas, which is fermentation, or they'll use a catabolic process where they make a protein, and the protein shows up as a basic molecule in the broth. So this is the entire range of the phenol red broth test. So the red or orange is neutral, the yellow is acidic, and the fuchsia is basic. Now I've manipulated those with a weak acid and a weak base, just so that you could see the difference in the pH range for the pH indicator. The phenol red is the most commonly used pH indicator in microbiology. In a practical, if I ask for a pH indicator and you don't know it, you should guess phenol red, because it is likely that's what it is. 60% of all the tests we use have phenol red in it. The others have different colors. They're blue and green or purple and yellow, they're different. But 60% of all the tests we use in microbiology use phenol red. Okay? So what you're gonna do with this today is on the back, behind Desiree, you'll see that there are three racks. One of them contains glucose, one of them contains sucrose, and one of them contains lactose. It could be any sugar. Those three are used because they're the most common sugars used to differentiate microorganisms. So not only are we talking about biochemical activity, but we are establishing our ability to differentiate results of tests, because every single organism that is on this planet has a different biochemical profile, and they will react to different substrates in different ways, And you can look at that biochemical profile and you can use it to identify the organism. That's what this book is about. Now, this book right here, we used to use it in the lab, but it's so cumbersome that I just made my own charts and tables, and I will give you those when we do unknowns in Unit 3. And the unknown will be exactly worth one practical. It's an oral exam, you and me looking at each other. I'm asking questions, you give me answers. Okay? You will be graded on process and ability to communicate results. I will watch everything. Okay? It's a checkoff. You're getting experience in checkoffs. It's gonna happen. Okay? By the way, most people love it. There are three different defenses of the unknown on my YouTube channel that you can watch. Okay? Good. So what you're going to do today is you're going to do everything by cohort today. So you'll need this setup. You'll need a setup of the carbohydrates. You'll need one glucose tube, one sucrose tube, and one lactose tube, right? Then you'll need 2 of the gelatin we haven't talked about yet, and then you need 1 of each of the plates. So there'll be 1 tributyrin, 1 skim milk agar, 1 starch agar, and 1 deionase agar. And you're going to do this as a group. That's why it's not going to take very long. Okay? These are all the same. I did that for a reason. Because if you go over there and pick the tubes up but don't label them, you're not going to know what they are. Even though Myra has actually used a different cap top color for the different sugars, you're still not going to remember. So when you go in there to pick them up, take a Sharpie and put an L for lactose, an S for sucrose, and a G for glucose. And each of the cohorts is just going to do 1 organism. Will you hand me one of those sheets? Thank you. So why don't we say that table 3, that's you, Christy. Why don't you all do Enterobacter orogenes, so the second one. Table 2, that's you all. Why don't you all do E. coli? Table 6, that's you, Itati. Why don't you all do alkalogenes fecalis? Why don't you all do Staph aureus, table 5, and table 4, why don't you all do Serratia marcescens, the very last one, okay? So you'll need 1 glucose, 1 sucrose, 1 lactose. You'll take the organism from the slant, put it into each of those broths aseptically, right? And you need to flame in between each one because you don't want to cross-contaminate with the different nutrients, okay? And then we'll come back and I'll explain the results, and then you can take pictures of all the different results. Every organism is going to have a different result profile. I don't expect you to memorize those. I expect you to know how to evaluate them. Okay? Anybody else have questions? Okay. We'll do gelatin now. This is what gelatin looks like. It's got gelatin in it. How many people like gummy bears? That's what it is. No sugar though. Just the gelatin. We're looking for the ability of a microorganism to utilize gelatin as a nutrient source. It's a semi-solid. It kind of has the consistency of Jell-O, although it's sticking really nicely right now. So it doesn't make a difference how you inoculate this. You just need to inoculate. You can use you can use a loop or you can use a needle. And you can like stab it all you want. If you're going to get some frustration out, you just want to stab it, that's fine. It doesn't make a difference because once we put this in the incubator at 37 degrees at at body temperature, it's going to become liquid. But the organism is going to grow over the next day or so, and whether or not it has the enzyme to break down gelatin, it will either break apart the molecule or it won't. So the way we test for that is we take these out and we put them in the refrigerator, 4 degrees Celsius, for about 30 minutes afterwards. And if it take them out of the refrigerator and it turns back to solid, it's negative. If you turn it over and it just moves a little bit, just any liquid at all, it's positive. Okay? Now we won't have to do that because Myra will come in tomorrow, and at the end of the day, she will move everything out of the incubators into the refrigerators so they don't continue to grow over the weekend. Okay? So whatever happened by tomorrow will be the results that you'll see on Tuesday. Okay? How many of these do you need? 2. Follow the link. So you're going to inoculate Bacillus cereus in one, and then you're going to do Staph aureus in another. Okay? Not yet. I got more. This is starch agar. Starch agar has got agar in it, and we've added starch, potato starch. So you can't see the starch. It's mixed in there very nicely. It's at 1%, I think, maybe less. Well, what we're gonna do here is we're gonna take the plate, we're gonna divide it into half. On one side, you're going to inoculate with Bacillus subtilis, and on the other side, you're gonna inoculate with Staph aureus. Now listen up. When you draw the line, will you please inoculate on the outer edge, not in the middle, kind of on the outer edge, because some of these organisms produce so much enzyme, it's gonna clear most of the plate. So if you do it on the outer edge, you'll get to see the true result of the other guy. You don't want them to run into each other and just clear up the entire plate. When exoenzymes are released from bacteria, they're released radially. What does that mean? In all directions. So the enzyme is going to be released in all directions. If it comes in contact with a substrate, it's going to bind the active site, and it's going to denature the substrate. And you're not going to have starch there anymore. You're going to have some glucose. Yeah. So we'll come back in 24 hours, and we'll flood the plate with iodine. Iodine is an indicator for starch. Where starch is, it will turn purple or dark. Depends on who makes it. Sometimes it looks really beautiful purple. Sometimes it looks black. It doesn't make a difference, it's dark. If it's dark around the colony after the addition of iodine, it's negative because starch is still there. If it's clear around the colony, that means that the organism produced the enzyme that broke down starchase, and starch is no longer there. So that would be positive. Okay? By cohort. Good? This is skim milk agar. What does it look like? Looks like it has milk in it, and it does. So Myra is really good at making media, so we save a lot of money. We're Riverside. We don't get a lot of money. We have to make our own stuff. HLC buys everything. But Myra is a pro at this stuff. So what she had to do is she had to autoclave the agar. She had to filter sterilize the milk. And then when the agar was cooled down almost to room temperature, she put the milk in it and mixed it up gently, and you have skim milk agar or casein agar. We're looking for the ability of the microorganism to break down the major protein in milk, which is casein. Not the sugar. The major sugar in milk is lactose. We're looking for the breakdown of casein. So what you're going to do here is divide the plate in half. On one side, you're going to inoculate with Bacillus subtilis, closer to the edge. On the other side, Alkalinogeni's faecalis. Okay? And then you'll just come back in 24, in our case, it'll be next Tuesday. If it's still milky, I didn't have a better name for milky. I could have said white, but milky sounded better. If it's milky around the colony, that means that the organism couldn't break down the casein and it's negative. If it's clear around the colony, that means that the organism broke down the casein and the organism is positive for the production of an enzyme called casease. Okay? Remember, A's at the end of anything means enzyme. Okay? Any questions? One of the prettier plates is this one. This is DNA agar, and we got DNA in it. So DNA, we're looking for the ability of a microorganism to break down DNA. We add a presence-absence indicator. It's not a pH indicator, which is methyl green. Methyl green is attracted to DNA. It's going to bind it. It's going to attach to it. So you're going to cut the plate in half, draw a line in the back, not in the front, in the back. On one side, you're going to inoculate with Enterobacter aerogenes. On the other side, you're going to inoculate with Serratia marcescens. Right? Come back in 24 hours. If it's still green around the colony, it's negative because the organism didn't break down the DNA, and the methyl green is still there, saying, I'm still here. If it's clear around the colony, that meant that the organism had the enzyme that breaks down DNA called DNase, and it's cleared, because if you break apart the DNA, the methyl green also disappears. Yeah. Pretty cool. All of these plates are enriched, and they're all differential. Right, we talked about that in class already. The last plate is called the lipase test. Now, it can be any lipid. We've selected a lipid called tributylate. Tributylin is a fairly long chain fatty acid. And so what we're looking for, does the organism have the ability to break down that fat? Now it could be any fat, but in this case it's tributylin. On a practical, if I say, well, what's the substrate in the lipid hydrolysis test? The answer is going to be tributylin. Okay? So what you're going to do here is divide the plate in half, and on the very end, you're going to inoculate Enterobacter aerogenes on one side. On the other end, you're going to inoculate Staphylococcus aureus. And you're just going to let it sit in the incubator for 1 day. That's going to be plenty of time. Staph aureus absolutely loves tributylin. It loves it. You're going to see Staph aureus on here, and it's going to be bright golden. It's going to be gorgeous. But the other really cool thing is, how many people like feta? If you like feta and you open this up, it's going to smell like feta, because the breaking down of that long chain fatty acid smells like the breakdown or the odor that feta gives off. It always makes me hungry. Even smelling it now makes me hungry, because it kind of smells like feta cheese now, but wait till it starts to break down. It smells better. That's for you. Does anybody have any questions? Yep.
I was just gonna clarify, so for the gelatin,
Hydrolysis?
yes, you said Staph aureus instead of E. coli. Is that
I
right?
did, no. Follow what's, sometimes I just jump to
Okay,
conclusions.
yes.
So it's E. coli. Hey guys, listen up. It's 12:34. Do you all have your answer with the antibiotics, or do you want to think about it more? Is every group okay with doing the project for the antibiotic and